A-level biology students, let's start on the kidneys by looking at their structure and then ultrafiltration in the glomerulus. A kidney has three main layers: the cortex, medulla and pelvis. The pelvis connects to the ureter, the tube that drains urine to the bladder. Spanning across the cortex and medulla are about 1 million tiny tubes called nephrons, surrounded by a dense capillary network. In this video we're going to zoom in on the first part of the nephron, the Bowman's capsule and the glomerulus, where ultrafiltration occurs. The glomerulus is a knot of capillaries, encased by the Bowman's capsule, which is the start of the nephron tubule. Ultrafiltration occurs because of high hydrostatic pressure in the blood. This is generated because the afferent arteriole, bringing blood into the glomerulus, has a wider lumen than the efferent arteriole, which takes the blood away again. Blood enters the glomerulus more easily than it leaves, so pressure builds up inside the capillaries. This high hydrostatic pressure forces small substances out of the blood. Water, urea, glucose, ions, and other substances like amino acids are pushed through the pores in the capillary endothelium. They then pass through the basement membrane and finally through gaps between specialised cells in the Bowman's capsule epithelium called podocytes. Large substances, such as large proteins and blood cells, remain in the capillaries because they are too large to pass through this filtration barrier. The fluid that collects in the Bowman's capsule is called the glomerular filtrate. So ultrafiltration is essentially a pressure-driven filtration process that separates small molecules from the larger components of the blood. This filtrate is the starting point for the next stages of urine formation in the nephron, and I'll be covering these over the coming weeks, so make sure you follow so you don't miss it.